Lesson 13, Prokaryotic cells. Cells fall into one of two broad categories, prokaryotic and eukaryotic. We classify only the predominantly single-celled organisms, bacteria and archaea, as prokaryotes. Animal cells, plants, fungi, and protists are all eukaryotes. All cells share four common components. 1. A plasma membrane, an outer covering that separates the cell's interior from its surrounding environment. 2. Cytoplasm, consisting of a jelly-like cytosol within the cell in which there are other cellular components. 3. DNA, the cell's genetic material, and 4. Ribosomes, which synthesize proteins. However, prokaryotes differ from eukaryotic cells in several ways. A prokaryote is a simple, mostly single-celled, unicellular organism that lacks a nucleus, or any other membrane-bound organelle. We will shortly come to see that this is significantly different in eukaryotes. Prokaryotic DNA is in the cell's central part, the nucleoid. Most prokaryotes have a peptidoglycan cell wall, and many have a polysaccharide capsule. The cell wall acts as an extra layer of protection, helps the cell maintain its shape, and prevents dehydration. The capsule enables the cell to attach to surfaces in its environment. Some prokaryotes have flagella, pili, or fimbriae. Flagella are used for locomotion. Pili exchange genetic material during conjugation, the process by which one bacterium transfers genetic material to another through direct contact. Bacteria use fimbriae to attach to a host cell. At 0.1 to 5.0 micrometers in diameter, prokaryotic cells are significantly smaller than eukaryotic cells, which have diameters ranging from 10 to 100 micrometers. The prokaryote's small size allows ions and organic molecules that enter them to quickly diffuse to other parts of the cell. Similarly, any waste produced within a prokaryotic cell can quickly diffuse. This is not the case in eukaryotic cells, which have developed different structural adaptations to enhance intracellular transport. Small size in general is necessary for all cells, whether prokaryotic or eukaryotic. Let's examine why that is so. First, we'll consider the area and volume of a typical cell. Not all cells are spherical in shape, but most tend to approximate a sphere. You may remember from your high school geometry course that the formula for the surface area of a sphere is 4 pi r squared, while the formula for its volume is 4/3 pi r cubed. Thus, as the radius of a cell increases, its surface area increases as the square of its radius, but its volume increases as the cube of its radius, much more rapidly. Therefore, as a cell increases in size, its surface area to volume ratio decreases. This same principle would apply if the cell had a cube shape. If the cell grows too large, the plasma membrane will not have sufficient surface area to support the rate of diffusion required for the increased volume. In other words, as a cell grows, it becomes less efficient. One way to become more efficient is to divide. Other ways are to increase surface area by foldings of the cell membrane, become flat or thin and elongated, or develop organelles that perform specific tasks. These adaptations lead to developing more sophisticated eukaryotic cells. In the next lesson, we will go into more detail about eukaryotic cells. See you then.